This is a short presentation to discuss a project that we're working on with HDRUK using multiomics to understand what is known as multimorbidity. And it's one of the ways of explaining how we're going to use big data to understand a complex problem. Multimorbidity is one of the UK's biggest health challenges, and all we mean by multimorbidity is having more than one health condition at the same time. And the health conditions associated with multimorbidity can be almost anything, but the most common ones are shown in this diagram here. And the larger the words, the more commonly associated it is with multimorbidity. And as I mentioned, multimorbidity is extremely common, with nearly a quarter of the population, or just over a quarter of the population, having more than one long-term health condition. And the older that people get, the more likely it is that they have multimorbidity, with nearly 2/3 of those aged over 65 having more than one health condition. It is also significantly associated with poverty, and the more deprived groups are, the more common or the more likely it is that they have multimorbidity, and also the earlier multimorbidity is likely to set in, so they get multiple health conditions more often and at an earlier age. With multiple conditions also comes multiple medications for many people, and we call this polypharmacy, and it can lead to all sorts of things like side effects where medications clash with one another. And the more medications you have, the more side effects you're likely to have. Multimorbidity also accounts for a significant proportion of healthcare usages, with almost 65% of outpatient appointments being used, or at least 65% of the people that would be seen at outpatient appointments having more than one healthcare condition. And of those that are in hospital, around 70% of those would also have more than one healthcare condition. And this results in significant costs for healthcare. One of the things that we would like to do is understand multimorbidity better, with finding better treatments for people that currently have multimorbidity, and ideally finding ways to prevent multimorbidity developing for people in the future. And to do this, we need to understand much more about the causes of multimorbidity. But this is quite complex because multimorbidity isn't a single condition, as I said earlier. It's lots of conditions in lots of people. So how can we even begin to untangle some of the causes of this particular group of conditions? We can start by asking ourselves, well, what is disease? Or equally, what is health? And one of the best ways that we have to understand health and disease better is to compare people that have a condition to people that don't, and look for what the differences are. And one of the tools that we have for looking for differences in biology are known as the omics tools. So these allow us to look for differences between different types of biological molecules in cells and organisms. These tools allow us to ask what kinds of molecules are present in a cell, and also how much of each type is present. And again, if we can compare cells from people that have a condition to cells from people that don't, then we can start to understand what about that particular condition is giving rise to those differences. The word ending omics is related to the word ending ome, which simply means the complete set of. So we can use omics tools to look for the complete set of biological molecules such as DNA, RNA, protein, sugar, fats, and more. And there are different omics for different omes. So one of the most common ones looks at the genetic information inside a cell. And we call this genomics. And this allows us to understand the genome, the complete set of DNA that is present inside of every cell. But we don't just stop there, because although genomics can give us lots of information about what could happen in a cell, because all cells have the same DNA, it doesn't necessarily tell us much about what of the DNA is being used. And to do this, we need tools that look at the so-called expressed genome. And the first message in the expressed genome is what is known as RNA. And RNA tells us about the genes that are actually being used in a particular cell or in response to a particular situation. So for example, we could look at what genes are expressed after a cell becomes infected with, for example, a virus. We can also look at the proteins inside a cell using the tool called proteomics to understand the proteome. And we can use a tool called metabolomics to look at metabolites in a cell and understand the metabolome. And most people think of metabolism as the way that we convert food into energy, but metabolism can be regarded as any kind of chemical change in the cell. So yes, we can look at sugars and the conversion of glucose with metabolomics, but any kind of conversion of a chemical substance. So we could also look at, for example, the conversion of drugs and medicines that people take using metabolomics. And it can tell us a lot about what's actually happening inside a cell. And all of these omics tools make up tiny pieces of a very complex puzzle. And in addition to the tools that I've described that look at DNA, RNA, protein, and metabolomics, there are a whole variety of other omics tools that can look at further parts of the cell in detail. And this includes things like lipids to look at fats, sugars, as I mentioned earlier. There's a branch of this science called immunomics, which looks at the immune system and how that varies. And yet, even with all these tiny pieces of the puzzle, there is still a missing piece. And this missing piece is the role of the environment. So we know, for example, that heritability, the genes that we have, only accounts for some experience of the disease. And we know this from things like twin studies, or the fact that even if you have a gene for something like cancer, it doesn't necessarily mean that you go on to develop the disease. And the rest of the reason that we get disease comes from what we call the environment. And by environment here, I don't necessarily mean oceans and trees and things like that. We mean the external influences that you experience as a person that might influence your health. So this includes things like your diet, what you eat or don't eat, how active or inactive you are, how clean the air that you breathe in, and whether or not you've been exposed to any kinds of drugs or chemicals. So things like smoking, like asbestos, like the amount of alcohol that you consume, all affect, or we call all of this the environment that you exist in. And researchers know that diseases are caused by a combination of the environment that you're in acting on the genes that you have, or in our case, diseases, since we're talking about multi-morbidity. And there is a final omics tool that we can use to investigate this, and it's known as phenomics. So if we know that diseases are caused by the environment acting upon the genes, and the way that we discuss this in research is we talk about your genotype, and that's the full set of DNA that you have, and how the environment acts upon that gives us the phenotype. And phenotype simply means what we actually see, the outcome of the environment acting upon your genes. And an example of this is something like eye color. So you might inherit a gene for blue eyes from your mother and brown eyes from your father. The actual shade of brown that your eyes turn out to be is your phenotype. So your genotype is blue and brown, those are the genes that you have, and your phenotype is light brown, that's the color of eye that you actually go on to develop. Other phenotypes are things like height, the diseases that you actually develop, whether or not you're lean or less lean. If you develop allergies, would be part of your phenotype, how intelligent you are, how fertile you are, and also how active or inactive you are. And the tool that we use to analyze these are phenomics. And to do this, we understand the phenome. And this can tell us quite a lot of information about health and disease, and particularly how the environment influences our genes to give rise to disease. So does that mean we can simply assemble all the pieces of omics to understand any given individual with multiomics? Well, would it be that simple? Because unfortunately, there are almost infinite pieces of this puzzle. If you consider that the human genome alone has more than 20,000 genes, and then that express genome that I talked about can vary dramatically between cell types and environmental conditions, the time of day that it's taken, whether or not you have an infection or indeed are undergoing a disease, gives rise to lots and lots of variation. The phenome is absolutely enormous. If you think about all the different phenotypes that a person can have, even just based on their appearance, let alone their experience of health and disease, then that's absolutely enormous. Likewise, the environmental exposures that you have undergone between your present age and when you were born are almost impossible to count up, and also can change over time. So, for example, most children, thankfully, don't smoke. You might start smoking in your teens and then stop again and pick it up later on. Or you might, your diet, for example, or your exercise levels vary dramatically throughout your lifetime. Omics tools can also be used on multiple sample types. So for a given individual, we might take a sample from their blood, from their urine, from their saliva, and from their tissue, which gives us 4 sets of all of the above information. And also, because we're particularly interested in changes over time, we might take samples for omics at multiple time points. And so it just increases the number of data points that we have to analyze in this system. So even just for a single individual, there are almost infinite data points. And the scale of information overall is huge because, as we mentioned earlier, we're talking about multiple people with multiple conditions and multiple data points for each person. So how can we possibly begin to untangle how all of this information is causing or contributing to the development of disease in different people? Well, we can use the omics tools, obviously, and we could design an ideal study that would help us to understand multimorbidity. We'd simply need to recruit some people. Well, thousands of people actually, because again, there's lots of different variations in people. We'd want to follow them from birth, or potentially actually a little bit before that, because we know that the environmental exposures that you have in the womb can contribute to disease development later on. We'd want to get multiple tissue samples from them and blood samples and saliva samples throughout their lifetime. And we'd also want them to fill out regular questionnaires and surveys about the exposures that they've had and the times they've been to hospital and whether or not they use any drugs. And then we'd want to follow them over time, so 20, 40, 60 years even, until the development of any kind of disease. And then we need to spend some time crunching the data, and hey presto, we'd get our answer. Simple? Well, yes, simple, but not particularly feasible, not least because it would be extremely expensive to follow hundreds of thousands of people for up to 60 years of time. So instead, let's think about a feasible omics study rather than an ideal omics study. And one of the best ways that we can do this is to make use of the data that we already have available to us. One of the really cool things about being in the UK is that we have multiple long-term, what we call longitudinal studies that have taken place with large subsets of the population. And these studies cover hundreds of thousands of people for more than 10 years of their lifetimes. We know lots of demographic information about the people that were represented in those studies. And for many of those studies, we already have some kind of omics information available. And not only that, but many of them also took and saved biological samples that we could do further omics analyses of if we wanted to. And the other thing that we are really lucky with in the UK is that we also have the NHS. And for many of the individuals in these studies, we have 50, 60 years of NHS records that we can also go back into to compare, of things like diagnoses that these people have had, medications that they've taken throughout their lifetime, times that they've been to hospital, and what their vital signs were in those studies. So by pooling all of this information, we can get the same kind of data that we would get if we started a study from scratch. Of course, we'll need a few more things. We will need some serious computing power to be able to crunch the sheer amount of data that will be involved here. We need some pretty advanced statistical methods in order to be able to understand the relationships between all the different variables. And we will definitely need some expertise and some people that can understand what the outputs of this particular statistical modeling will even mean. And to do all of this, we have the molecular studies of multiple conditions, the Multiomics consortium from Health Data Research UK, of which Cambridge is a leading player. And what this makes use of is 9 population studies that have already taken place in the UK, covering more than 750,000 people and more than 10 years of data with the NHS health records. And there are 2 purposes to this study. The first of which is actually just about feasibility. So when these original data sets were collected, they weren't designed they weren't collected and designed in such a way to make it easy to pull them all later on. They were designed for the individual studies. And likewise, health data that is collected by the NHS is collected for people's healthcare, not for research purposes. So it's all saved in different locations and written down in different ways. And so the first goal of this study is just to see whether or not it's even possible to be able to combine all the different pieces of information in the same place in a way that is usable and analyzable for researchers. And if that is possible, and we do believe that it is possible, although we will need to develop some new tools and techniques to do so, then we want to use all of that information to be able to look for links between the studied biology, the environment, and the diagnoses. What kind of information might a study like this find? Well, it's not likely to completely unravel the causes of multimorbidity, as I'm sure you can imagine. It might get us to a state where we understand quite a bit more about multimorbidity, which is what we can really hope for. So something that looks more like this, or more likely something that looks more like this, which thankfully multiomics and data specialists can understand and make some meaning for that, because we're looking for connections between different biological outcomes and disease and environment. What does this mean for actual people? Well, the types of things that we expect this kind of study will help us to unravel are things like if we have 30 years of data and samples and we know a person develops high blood pressure at age 60, we might be able to look back through their samples and look for changes within cells and tissues, so for example proteins or RNA, that warned us about the onset later on, so we could use it for early treatment or prevention. We hope that it will give us a lot of information about what combination of genetics and DNA and environmental exposures leads to disease. So why is it that people with a particular genetics go on to develop cancer or not, or diabetes or not? And is there a way that we can use that information to prevent more people developing those conditions? We think it will help us to understand the basis behind different forms of the same disease, so for example inflammatory bowel disease, that look very similar but actually have very different causes. So we might be able to find more appropriate treatments for individuals with the same symptoms that actually match the causes of their conditions. Conversely, we expect it will show that some diseases that look really quite different actually have similar causes. So for example, things like high blood pressure and diabetes, we might be able to target a single pathway in order to improve people's symptoms there. We also hope that it might help us to prevent the onset of things like multimorbidity by following diagnosis or changes that happen in cells and the things that we're measuring after one diagnosis that let us have warning about the potential development of a new diagnosis so that we can prevent the onset of multimorbidity. And the really exciting thing about this particular study and the use of health data for research purposes like this is that unlike if we had to start from scratch and develop sort of the so-called perfect study that might take 60 years in order to give us the amount of information that we need, studies using health data, because the data is already collected, can give us information and answers in a much faster time frame. So we're looking at more like 5 years potentially, or potentially even faster, in order to be able to influence healthcare. And what we're in the process of doing at the moment with you all for patient public involvement is working with people like yourself and people that were involved in these original studies to help share with them what we're doing with the data now and why it might be useful in the future. Thank you.